I'll just start sharing here. Can you see my screen?
Yes, sir.
Yes, sir.
Okay.
Yes, sir.
Just to check, pointer to the left,
Yes, sir.
top,
Yes, sir.
to the right,
Yes, sir.
and to the bottom here.
Yes, sir.
Okay, so we are meeting after a while here. Uh, and one of the main reasons is much of the portion that we had to cover is actually done. And we also had our final year exams going on. Uh, and they should also be done this week. Okay, so there's one topic towards the end which I thought we had not covered in detail, and that is how gene expression is regulated in organisms. Okay, and an example of gene regulation is what we refer to as the Lac Operon. Okay, so we'll see what that is. Many of you probably have a little bit of an idea of what that Lac Operon is based on some of the classes you have had earlier, maybe in your school, high school, as well as in college. Anyone has heard of this Lac Operon? Anyone familiar with Lac Operon? You can unmute and talk if you
Yes.
wish. Yeah, say something. And
Sir, it is a bacteria usually uses glucose as a source of energy, but in absence of glucose and presence of lactose, it can still grow using lac operon mechanism.
Lac Operon mechanism. Who was this? Say your
Sir,
name, maybe
Ankit.
Ankit. Okay. Anybody else want to say something about Lac Operon, which they recall or they
So
remember?
Sir,
it
there
is
are
based
three
on
enzymes as well.
lactose. It is
Acha.
basically in bacteria for the metabolism and transport of lactose. It is
Okay.
somewhat something related to that.
Something related to
Is
that.
a repressive type of Lac Operon which regulates the metabolism of lactose. And there are three genes Z, Y, A, which
Uh,
control its metabolism, lactose metabolism.
okay. Anyone else want to say anything? Okay. Uh, Almost everything that you have said seems to be connected somewhere correctly to what is Lac Operon is. But we'll look at the fine details as we go along. Okay. So we all know, for example, that in most of the biological systems on Earth, as you know, DNA is the genetic material which is a repository of all information that is required for the functioning of an organism. And generally, DNA has two functions. One which is called as replicative function, where it is it's copied faithfully uh, to another molecule of DNA as part of what is called a cell division process, so that the genetic material can be passed on to the progeny. The other role of DNA is the carrier of information, and the information of DNA is decoded through two processes. One, which is the process of transcription, which converts DNA to a RNA message, as it is called, and then the process of translation, which reads the message in the RNA and converts it to a protein product. And it is these proteins many times which carry out many other functions in the cell. Likewise, in some cases, the information content in the DNA might be the RNA molecule itself. For example, as you have seen in case of tRNA or rRNA and so on and so forth. Okay, so the message in the DNA is in the form of a product which could be RNA or a product which could be a protein, and this is a typical information flow which you call as a central dogma. Now, what you should also realize is that different gene products, okay, that is along the DNA where there are different sequences which we call as genes, the product of these genes have different functions. Okay, so some of the gene products, as I said, are RNA molecules which carry out very specific functions. For example, ribosomal RNA takes part in synthesis of proteins. Then tRNA acts as a little message to help ribosomal RNA do its job of translation. The other gene products could be protein itself, like let's say insulin or actin or myosin, and they all have their own functions. And the other aspect is that these gene products, which have different functions, are needed by the cell at different times. Okay? Some are needed once in a while, some are needed all the time, some are needed like in large amounts, in spurts, and so on and so forth. And then therefore, the situation of which gene product is needed when is unique to a given cell and therefore to a given tissue or a given organ, uh, and is a function of a lot of regulatory issues. Okay? Now, as I said, some gene products are needed all the time. Okay, for example, a protein which does some function in the cell, which is occurring all through the life of the cell. Okay, and such genes which code for certain uh, proteins which have these type of all 24/7 function are called as housekeeping genes or housekeeping proteins. Okay, and since they are expressed all the time, that is, the gene is being transcribed to RNA and the RNA is being translated to protein round the clock, this type of genes are said to have what is called as constitutive expression. That is, they get expressed all the time at a basal level. In other cases, what you find is that there are some genes which are produced, which are expressed at a certain rate, and periodically they need to be shut down, or periodically they need to be overproduced. Okay, so these type of systems where periodically you need to stop what the gene is doing in terms of giving the RNA and the protein, and that process is referred to as repression of gene expression. And when the gene is kind of triggered a little bit or activated so that more RNA is made and more protein is made, such a phenomena is referred to as induction of gene expression. Okay, so you actually have basically three situations in a cell going on at any given time. One is a constitutive expression of some genes, which gives you what are called as housekeeping proteins or what you call as housekeeping genes. And there are other systems where some genes are being repressed because at that point their expression is not required, or at a given point DNA is being genes are being induced and they are expressed to at a faster rate because there's more need for the protein product of that gene. Okay? Now, because of this situation, you need what is called a gene regulation. Okay, if you do not regulate, what will happen is the RNA, which is sometimes the product of the gene or the protein, will end up being made indiscriminately. Okay? In other words, they'll be just made kind of at random, and because of that, you'll find that sometimes there will be scarcity of some RNA or protein, and in that case, the cell function will be compromised. Or in other cases, because the production is indiscriminate, you might just overproduce something, and because of that, you might have cell function issues. Again, the functions of the cell might be affected because something is present in a large amount, more than what is necessary. Also, you might have energy wastage because every time you make a protein, it requires ATP to join amino acids together. So if you're just making protein when it's not needed, it is energy wastage. And likewise, you might be wasting resources. That is what you call as amino acids, etc., which are crucial. And sometimes, if you remember, certain amino acids are essential amino acids. You cannot produce it. You need it in a diet. So if I need something in a diet, and therefore it is in a short supply, and I start wasting it, then there'll be a problem. Okay? So this aspect that genes need to be regulated so that the cell functions correctly was kind of known for was part of the thought process for a while. Okay, because people realized that for a cell to function very correctly, you need the DNA message to be read in a very systematic fashion. In addition, people had observed that, in many cases, under certain circumstances, there was cellular changes occurring in response to external stimuli, indicating that there is some sort of regulation of what is called as gene expression, or how genes are transcribed into RNA and finally into protein. So, one of the first ideas of how genes might be regulated, or how in a cell production of RNA and protein might be controlled, was the work of Francois Jacob and Jacques Monod way back in 1961. Okay, both people who are working in, I think, if I remember right, in the Pasteur Institute, along with a third gentleman, Andrei Lvov. Okay, all three of them were working on projects which are very similar in terms of what we call as how cells regulate the expression of their genes. Okay, and uh, the initial idea was that much of the regulation is probably occurring at the level of transcription, where I control how the DNA is actually converted to RNA. Okay? And if I, what you call as trigger this process such that DNA is read more vigorously to give you a lot more RNA, then that process is referred to as induction. And at the same time, if the production of RNA is being halted or being reduced, then that type of regulation is referred to as repression. Okay? And this idea slowly gained, momentum, and terms like negative and positive regulation started being, used. A negative regulation is where the presence of an effector, okay, and effector means some factor results in reduction in transcription. So that type of regulation is called as negative regulation, where because of the presence of a control element, you have reduction in the transcription. Okay, and these effectors were called as repressors or inhibitors. Likewise, you could also have what is called as positive gene regulation, where the presence of a effector molecule or an entity, whatever that might be, results in increased transcription. So these entities are then referred to as inducers or activators. Okay, and for this idea that and showing how regulation actually occurs in E. coli in that time, and in one of the viruses, which switches between what is called as lysogenic and lytic phases. Jacques, Monod, Jacob, and Andre Wolf collectively got the Nobel in 1965. Okay, dealing for throwing light on what is called as gene regulation, and they are the ones who they introduce the concept of what is a operon. Okay, and the operon was defined as a unit of the bacterial expression and bacterial gene expression and regulation, which includes what were referred to as structural genes and control elements in DNA that were recognized by regulator gene products. Okay, now this is what we call as effectors. So these effectors are somehow affecting the DNA at points which are referred to control elements, and because the interaction of the effector at the level of the control element, we have what is called as expression increase or decrease of what are called as structural genes, that is the target genes. Okay, to elaborate this further, is one as a regulator gene. So the function of this regulator gene is to have a gene that codes for a product, usually a protein, that controls the expression of other genes, usually at the level of transcription. So the idea of this regulator gene is shown here. So this gene, when it itself is transcribed, gives you RNA, and then that RNA is translated to give you a protein, and this protein is referred to as effector protein or regulator protein, and what it does is it binds in front of another gene sequence which is related to the target gene, what we call as a structural gene. And when this regulator binds to the DNA near the site of where the structural gene is, it can alter the way this gene will be expressed. Either it can increase the expression or it can decrease the expression. And this gene whose expression is affected is referred to as a structural gene. That is a gene which codes for any RNA product other than what you call as a regulator. Okay, so the idea is the product of this gene is something which has got a cellular function different from this regulatory function. Okay, and then this concept of what is called as negative regulation and positive regulation. Okay, so that is shown here. Okay, so for example, let us say this is a sequence of DNA as part of the genome where there is a structural gene or what is called a target gene, in front of which there is a sequence where a promoter, which is called the promoter sequence or the operator sequence. Okay, so normally what happens is uh, at some point somewhere near this structural gene and the promoter operator site is another sequence which when it is transcribed and translated gives you a protein, and this protein sits at this site which is called as the operator site, or generally somewhere overlapping with the promoter and the operator site, and when that sits there, there is no expression of this gene. Okay, so that is referred to as a negative control. That means when the repressor is present, you actually have reduction in gene expression. Okay, as opposed to that, look at this situation here. Okay, so now what happens is normally when the protein is sitting here, it is turned off. Okay, and nothing is happening. Okay, and then again in this case also there's a sequence of a regulatory gene which when transcribed and translated gives you a protein. And when this protein sits at the same operator site, what happens is it enhances gene expression. Okay, so now you get more of RNA and more of that protein. Okay, so this type of control where binding of a effector results in enhanced synthesis of RNA and protein is referred as positive regulation. Okay, so in a positive regulation, a transcriptor factor is required to bind to the promoter to enable RNA polymerase to initiate transcription. Okay, and here what happens? A repressor stays all the time to do expression. So this is called as negative control, and this is positive control. Okay, so to reiterate
So
again,
what is
negative.
cis
Hello, hello.
Hello, sir.
Yeah, yeah, yeah.
Hello. So what is
Yeah,
cis
yeah, I can hear you.
acting site in the previous slide?
Uh, oh, uh, see, usually there are two aspects to what we call as regulation. One, what is called as cis-acting regulatory elements, and other which are called as trans-acting regulatory elements. Generally, to keep it very simple, cis-acting regulatory elements are elements which are part of the same genetic unit, as you can say, okay, or regulatory unit. So they're usually present close to where the target gene is and where the target gene's promoters and operators are present. And because they're present close by, they're affecting DNA which is present really close as part of the same system. Okay, and those type of control elements are called as cis-acting control elements. Sometimes what is also found is that you can have a system whereby this repressor protein which we are talking about or activator protein are present far away, and then their product actually floats and goes somewhere else on the DNA and binds and does its job. When you have a lot of distance between the regulatory element and the actual gene, then that type of control is referred to as a trans control. Okay, so that means generally something which acts as at a longer distance. And generally what is found is trans-acting things are proteins in nature, because you can have a protein which can go everywhere and bind elsewhere on the DNA and control the expression. And cis-acting elements can generally be proteins or even RNA because they act very close by. Okay, does that make sense? So I'm answering very simply. Actually, the answer is fairly complex in terms of what is cis and trans. Generally, it means cis acting elements are elements which are close to site which is being controlled. Okay, is that clear? So, as I said, likewise, you can also have expression regulation or gene regulation at the level of translation. Okay, now the examples I've given here are all occurring at the level of transcription, where I'm controlling the production of RNA and not worrying too much about how RNA is getting converted to the protein. But you can also have control of gene level, gene regulation at the level of translation. So what happens here in this case, if I have what is called the effector, which is a negative regulator, then in the presence of that effector, translation is reduced. The mRNA is not read correctly by the ribosomes, and I get reduced production of protein, and that again is referred to as a repressor or a inhibitor. Likewise, I can have something which is positive, where if I have an effector, then that results in enhanced translation, and that is generally referred to as activator. Okay. Now, in eukaryotic systems and also in some prokaryotic systems, things can get even more complex, where more than one entity can do this job of reduction or increase in transcription or translation. And when there are multiple agents which are taking part in this process, then those other agents are referred to as co-repressors or co-inhibitors or co-inducers or co-activators. Okay, and this is not uncommon. Sometimes you might have 2 or 3 or sometimes even 4 elements working together to control the level of regular expression of a certain gene at either the transcriptional level or at the level of translation. Okay, but let us look a little more detail on the Lac Operon, which was actually discovered or elucidated by Jacques and Monod. Okay, so the Lac Operon basically is composed of, in a sense, 6 elements. Okay, there is what is called as a Lac-I gene. Okay, and this Lac-I gene codes for an entity which is called as a repressor. Okay, the second is what you call as a Lac-Z gene or the Lac-Z gene, whose product is a protein which is called as beta galactosidase. The third is what you call as Lac-Y gene, and the product of that Lac-Y gene, when it is transcribed and translated, results in a protein which is called as Lac permease. The fourth is what is called as Lac-A gene, and the product of this gene is an enzyme which is a trans-acetylase. Okay. Now look at this. Whenever you talk about genes, that you should always write it in italics. So when you write in italics, it implies that you're talking about genes. And when you talk about the product of the gene, you always write it in regular phase. So you always, this is indicating they are talking about the protein. Okay. And in addition to these 4 elements, there are what are called as promoter sequences of the DNA and very specific operator sequences on the DNA. Okay, and the architecture of these 6 elements is somewhat like this in what we call the E. coli DNA. Okay, so if I look at the stretch of DNA along the E. coli genome, I find that the structural elements are what? The genes which are controlled, okay, are Lac-Z, Lac-Y, and Lac-A, which are present, what you call as downstream of what are referred to as promoter and promoter operator sequences. Okay, and here is that Lac-I gene. Okay, now see what happens. Lac-I gene, when it is transcribed and translated, codes for this repressor. Lac-Z, when it is transcribed and translated, codes for galactosidase. Lac-Y, transcribed, translated, codes for permease. And Lac-A, when it is transcribed and translated, codes for trans-acetylase. Okay. Now, because once started, all of these will be expressed at the same time. Such units are referred to as polycystonic units, or the mRNA that you get is referred to as a polycystonic mRNA. Okay, later on, when that mRNA gets converted into protein, you can simply cut those proteins and get 3 functional proteins. And this is very common in prokaryotic systems. Generally, such polycystonic mRNA is not produced in eukaryotes, and if so, it's very, very rare. Okay. The advantage of this is because all of them are associated with the same function of metabolizing galactose, they are either produced all together or none at all, and that allows you to regulate things much more easily and simply. Okay. Now, this Lac Operon, which we talk about in terms of classification, is referred to as a negative inducible operon. Okay, so what does this mean when you say negative inducible operon? Okay, so in a resting state, as it is called, when nothing is happening, what you find is that the product of LAC-I, okay, which is this repressor protein, is produced at a basal level, and it sits on the promoter or the operator side, and because it is sitting there, it doesn't allow the expression of what we call as LAC-Z, LAC-Y, and LAC-A products. Okay, and in that sense, the repressor is acting in a negative fashion. Okay, but the word inducible implies that in the presence of lactose, what happens is this repressor falls off, and suddenly the production of all the 3 products of LAC-Z, Y, and A occurs in a spurt. Okay, and because of that, we use the word inducible. So at the basal level, it has got a negative effect, but in the presence of the effector, you get what is called induction of expression or increase in expression. Okay, and this is shown here. So when you do an expression and you're say looking at the level of lac mRNA, okay, or the level of this protein which is called as beta galactosidase, as you add lactose in the medium, see what happens at the basal level. There is very little of lac mRNA. There is very little of beta galactosidase, and see what happens as soon as you add the inducer. Okay, so this is the point of adding the inducer. Inducer is what? In this case, lactose. And you find that very shortly, okay, just maybe even less than a minute, suddenly the levels of mRNA start to rise. Okay, and as they start rising, you'll find that closely paralleling it is also the increase in the level of galactosidase. Okay, and the reason for this slight time delay is, if you remember, every time I have a gene, I have to first make the RNA, and then from the RNA only I have to make the protein. And because RNA production is necessary chronologically before the production of the protein product, initial increase is always in the RNA, closely followed by increase in the protein level. So you see this slight off by a minute and a half or so. So here what starts almost at time 0. As soon as you add the inducer, here it starts at around a minute and a half. Okay, and as later on when you remove the inducer, again this falls off. Okay, and then for a while you find that the level of expression of the proteins, that is galactosidase and permease, continues, and later also this also falls off, and that also becomes to the basal. Okay, so now let us look at the details. Okay, I talked about all of them. So this LAC-I, which I talked about, as I said, is converted via mRNA to a protein which is called a repressor. Now this repressor is very interesting. Initially, you produce a monomer, and 4 of these monomers come together to form a tetramer, and it's a tetramer which actually binds at this promoter operator sequence. And when it is sitting there, it doesn't allow RNA polymerase to read this message and make mRNA. Okay, and that's why it is referred to as a negative regulator. Okay, the other product, which is galactoside permease, okay, and that is shown here as a product of LAC-Y. The function of that is to allow lactose cell very nicely and quickly, because normally lactose being polar will not enter the E. coli, and there is and the cell membrane doesn't allow entry of these molecules inside the cell. Now, once inside the cell, the lactose is subject to the action of what is called as galactosidase, which is a product of lac Z. Okay, and what does galactosidase do? it actually does two things. One, it takes lactose, breaks it down to the individual galactose and glucose entities. But it also does something very interesting. It also takes lactose and converts it into what is called as allolactose. And what is allolactose? Allolactose is actually an isomer of lactose where instead of having a beta 1,4 connection, you have what is called as a beta 1,6 connection between galactose and glucose. So essentially what has happened is what is called as transglycosylation. So the connection points have changed. Instead of being beta 1,4, there is now beta 1,6, and that entity is referred to as allolactose. Okay. So to put it all together, this permease allows galactose to come in. Galactose comes in. Once it comes in, galactosidase does two things: converts it into galactose and glucose. Then it can go into the TCA cycle and glycolysis and all those things. And it also converts it into allolactose. Okay. Now remember, all these are produced at a baseline level, very low levels when there is no lactose. Okay. Uh, uh, but because of that, what you find is in E. coli, even when there is no lactose at all, there's a very small amount of galactose permease which is being produced. There's a very small amount of galactosidase which is produced, and there's also a very small amount of the trans-acetylase which is produced. Now, interestingly, even today, we really don't know what is the real function of the trans-acetylase, but for some reason, it is also always located along with these other two. Okay, so imagine it's been around 60 years since we discovered this, but we still don't know what that trans-acetylase does. Okay, but see how they all work together. Okay, now remember, normally when E. coli is sitting there and there is no lactose, very small amounts of permease and galactosidase are produced at what are referred to as baseline levels. Okay, and because of this situation, normally what is happening is you have lac I, which is being read converted to mRNA. You produce that repressor, it becomes a tetramer, and it goes binds to the operator. And when it's sitting here at the operator site, it doesn't allow the expression of this. So because RNA polymerase cannot make the RNA. And this happens when lactose is absent. Okay, so repressor is active and the operon is in the off state. Off state means there is no production of RNA of lac Z, lac Y, or lac A, and therefore no protein. Now see what happens when lactose is added into the medium. Remember that little baseline level of permease and galactosidase which are there that allow that lactose to come inside the cell. Very small amount. And when the small amount of lactose comes in, this galactosidase converts it 50-50. In one case to lactose to galactose and glucose, and in the other case to allolactose. Now see what that allolactose does. Allolactose has a tendency to bind to this effector, which is a tetramer, and when it is bound to this effector, then this repressor is unable to bind at the site of this operator. So what happens is, in the presence of lactose, when the small amount of allolactose is produced, the repressor now falls off from that operator site. Now RNA polymerase is able to go right through and convert this lac Z, Y, and Z to the RNA, which is then translated to give you galactosidase, permease, and transacetylase. And this happens in a minute or two, and immediately the cell is able to take up galactose, I should say take up lactose, break it down to galactose and glucose, and start using it through its glycolysis cycle and TCA cycle. Okay. Now, there is an interplay between this lac operon and glucose. Okay, and because the reason is, remember, glucose is a normal source of energy for many of these cells as you try to grow them in culture. Okay, now glucose can modulate this lac operon, and see how it does so. Okay, now normally ATP is converted to ADP and also cyclic AMP, and the conversion to cyclic AMP is catalyzed by adenyl cyclase. Okay, now glucose inhibits adenyl cyclase, and because it inhibits adenyl cyclase, what happens is when glucose is present, there is very small amounts of CAMP because this enzyme is being inhibited. Okay. Now, when you reduce the level of CAMP, what it does is it reduces the binding of a protein which is called as catabolite activator protein, which is called as CAP. Now, what is the function of CAP normally? Now, CAP normally binds upstream of the Lac promoter. That means in the same Lac Operon, it binds further up, okay, somewhere even ahead of that Lac I sequence. And when it sits there, it induces the expression of all the genes of the Lac Operon. Okay. Now, see what happens when glucose is present. When glucose is present, what happens? It inhibits adenyl cyclase. When it inhibits adenyl cyclase, it reduces the level of CAMP. And when CAMP levels are low, this CAP cannot bind to that site. And therefore, if it doesn't bind to that site, there is no expression of the LAC genes. Okay. So, in a sense, glucose, when it is present, doesn't allow the expression of the LAC. Okay. Now, see the consequence of this. You can have actually 4 situations. Okay. One, there is only glucose in the medium and no lactose. Okay. Second, glucose is present and also lactose is present. Third is glucose is absent and also lactose is absent. And the last is glucose is absent and lactose is present. And see what happens to this operon in all those situations. Let us look at the first situation where glucose is present. Okay. So when glucose is present, see what happens. Remember, it inhibits adenyl cyclase, so you have reduced CMP, reduced cap binding, and low expression of LAC genes. So what happens is when glucose is present, doesn't matter whether there is lactose present or absent, you will not have any expression of the lactose metabolizing genes so long as glucose is present. Okay, but see what happens when glucose is absent. See, when glucose is absent, something very interesting happens. Now that cap protein starts to bind, right, to that. And when cap protein binds to that operon, it is going to enhance the expression of all the genes which are downstream, which includes LAC-Z gene. But remember one thing, when there is no lactose, that LAC-I is producing a repressor which is also sitting there. Okay, and when that repressor is sitting there, there will be no expression of LAC-Z genes because the repressor is having a high affinity for the site in the absence of lactose. But see what happens when glucose is absent and then lactose is present. Now what happens is this lactose gets converted to allolactose, and that allolactose removes the repressor. Again, remember, cap protein is still binding. That is also allowing the expression to occur very well. Now everything is put in place and all the things get expressed. Okay. So the important thing to remember is if glucose is present, it doesn't matter whether there is lactose is there or not, you will not produce the genes for lactose metabolism. If glucose is absent and there is no lactose, there is no need to produce any of those products, so they are not produced. Okay, and why they are not produced? Because the repressor is still sitting. And when glucose is absent and lactose is present, and that allolactose removes the repressor, and you start the expression of all the proteins. Okay, so this is what we meant. When there is no glucose, then ATP to CAMP conversion occurs to a greater extent. Okay, uh, uh, because CAMP is produced and CAMP and CAP sit together and it becomes active and you get good expression. Okay, and what I just said is kind of summarized here. Okay, so when you have no inducer, and what is the inducer? In this case, it is allolactose. Then the repressor sits there. We have no expression of these genes, and excuse me, and you do not get any production of galactosidase or permease or transacetylase. But when the inducer is present, what is the inducer? Allolactose. That allolactose binds to the repressor, and now the repressor cannot sit at this site. It falls off, and then you have enhanced production of the mRNA, and then production of the protein, which is coded for by the mRNA to give you galactosidase, permease, and transacetylase. Okay. Now, there are other very interesting systems for regulation, which are also, slight variations of just what I told you. And therefore, you have systems which are called negative inducible, negative repressible, positive inducible, positive repressible, and so on and so forth. Okay. But what is interesting is this is a picture of that Nobel Prize which was awarded in 1965. Okay. So, uh, if you look very carefully here, this second, third, and fourth. Okay. Uh, let's say second, third, and fourth. Okay. Second, third, and fourth on the right are Monod, Leboeuf, and Jacob. Okay. Monod, Leboeuf, and Jacob. Okay. And right next to them are Richard Feynman and Julian Schweinsteiger, who got the Nobel Prize in Physics at that time. Here is Woodward, who is also a Nobel Prize recipient in chemistry. And on the right here is also Nobel Prize recipient Mikhail. Okay. But see what is interesting. When Monod and Jacob talked about this, repressor and all these things, at that time, they didn't know what the repressor was made up of. Okay? They didn't know whether it was a protein or a RNA. And they initially thought it was probably RNA because it was easier to see that RNA can recognize DNA sequences and hybridize with it. And then by hybridizing, it will prevent the expression of the DNA. Okay. It's only that about 5 years later that Gilbert and Tashne from Harvard showed that these repressors are all protein in nature. Okay. But interestingly, they work exactly as Jacob and Monod hypothesized or talked about. But this idea of Monod and Jacob was actually not too far-fetched, because later, around 2000, it was shown that you can have regulation where you can control the expression of a gene by having RNA. Okay, and usually what happens is, if say this is a sequence of double-stranded DNA which is being transcribed to give you pre-RNA, then RNA, and then this RNA is translated to give you protein. If I were to read the opposite strand at the same region, what I will get is an RNA which is exactly complementary to this RNA. And if I process this RNA in a very systematic fashion, using, in this case, what is called as dicers, I will get a small piece of RNA which can actually complementary bind to this mRNA. And when it is bound to that, it might not allow translation to occur. Okay. And such type of control of how translation is done at the level of mRNA to protein, what is called as process, is referred to as RNA interference. Okay, which you now know, what you call a silencing RNA. Okay. Now, a similar idea is what we see in what is called as CRISPR technology. In CRISPR technology, also, if you realize that we talk about what we call as clustered repeats. Okay, and we talked about this at another this thing, but I'm going to just quickly tell you what this CRISPR technology is. Okay, so in CRISPR system, what happens is, in bacteria, when viruses infect a bacterium, what the bacteria does is it uses this proteins which are called as Cas proteins and degrades the viral DNA into small pieces. Then it does something very interesting. It takes these small pieces and puts it into its own genome at this region which are called as the CRISPR regions. Okay, the reason they are called as CRISPR regions is because in those regions you have what are called as palindromic sequences which are repeated, and in between them there are spaces which are unique DNA, which have got sequences which are different from this repeat. So what this Cas does is takes this piece of DNA and puts it in between this palindromic sequences to form new spaces. Okay. In that regard, now what the bacteria is actually doing is actually taking different pieces and inserting it at different location to give different spacers. And then later on, what happens is when this virus infects the bacteria again, then the bacteria goes to this sequences which we had already seen before. It transcribes this region, and when it transcribes this region, it produces short pieces of RNA, and some of this RNA are complementary to this DNA which the virus is introducing. And these RNAs then go and base pair with that DNA. And now the architecture of this RNA, which is called as CRISPR RNA, is such that it is able to again recruit a protein which is called as Cas9. And when Cas9 sees that there is complementary base pairing between these two, it just cuts that viral DNA and degrades the viral DNA. And because of this, now the bacteria is able to not get infected by this phage or a bacteriophage or bacteriophage. Okay, so this is the way the bacteria actually develops immunity. And the beautiful thing is the different viruses come in, different pieces are put here. In other words, the bacteria can adapt, and therefore this is also called as adaptive immunity. We also do this, okay, but we do this slightly differently. If you remember, when viruses infect us, we don't do anything to the DNA, but what we tend to do is we process the viral proteins and make it into small pieces and make antibodies to those antigens. Okay, but bacteria is doing it in slightly different fashion. They are actually making sequences which are complementary to the viral genetic material, and by that complementarity based binding, they are degrading the viral genetic material. Okay. And that is also shown here. So a virus comes in, all the different pieces of the viruses are put here as part of this CRISPR array, as it is called. And later on, when the virus actually infects, they will be expressed. And when they are expressed, you get different CRISPR RNA. Some of them will have complementarity to the infective agent. They will bind there, will end up cutting the viral DNA and therefore prevent infection. Okay, so the difference is that our immunity, where we produce antigens and antibodies, we have memory cells, but we can never pass it on to the progeny. But in E. coli, very interestingly, because it has become part of the genome, and the bacteria divides and passes on that genome to the daughter cell, or the daughter cell also has all that information about that viruses which have come. So in case of bacteria, this type of adaptive immunity is actually inheritable. Okay? Now, in case of this CRISPR technology, what the people who are working with this CRISPR thought was, if what we can do is modulate the system whereby I actually make this CRISPR RNA in a laboratory, okay, and put a sequence that I wish to put, then what I can do is, using this sequence which is under my control, I can target very specific DNA which are complementary to that sequence, and using this Cas9 protein, cut the DNA exactly where I want. Okay, and that is what is referred to as uh, CRISPR-Cas based genome editing. So in CRISPR-Cas based genome editing, what I'm doing is I'm doing what bacteria are doing, but slightly differently. Instead of waiting for some random event to occur in a bacterium and it making CRISPR RNA, I'm actually making this RNA in the laboratory by putting the sequence I want, which is complementary to the gene I want to cut. Okay, and that technology of what is called as CRISPR-based gene editing is what won the Nobel Prize. Okay, and the Nobel Prize was given just a week ago to Emmanuel Charpentier and Jennifer Doudna for this programmable dual RNA. Okay, so it's an extension actually of gene regulation, where the idea of gene regulation was taken forward and used for what is called as genome editing. Okay, so very interesting. What started off as a Lac Operon, an idea of Jacques and Monod, actually ends up today as a Nobel Prize winning idea, but for a purpose slightly different than gene regulation. Although bacteria use it for gene regulation, they are using it for a different purpose, for what is called as genome editing. Okay, so I'm going to stop sharing here, and I'm going to the
Hello, sir.
yeah. Okay.
Sir, can you explain again that dicers wala slide again? Where
Okay. 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 Let me just a second. I just okay. This one, no? This one.
yes, sir.
Okay. So now see what happens in this dicer technology or dicer system. Okay. So let us go to this part here. So this is normal double-stranded DNA, and in this double-stranded DNA, one of the strands is going to be read to give you what is called as the RNA. Okay, and remember that will be what is called the template strand. Okay, so when the template strand is read and you produce the RNA, you get first pre-RNA, and then if you remember the cytosol. In the cytosol, ribosomes come in, they read the message on this RNA, and you produce a protein. Okay. Now, one way to regulate how much protein is being made here is by using what is called as microRNA interference. So, in that, what happens is in the same organism, uh, typically the other strand is read as part of the regulation, and when the other strand is read, what will happen is you'll get an RNA which is exactly complementary to this. Okay? Now that complementary RNA is processed through what is called as this dicer. Okay, and what this dicer does is it processes this RNA into short pieces and gives you a small piece which is called as a microRNA. Now, since this entire RNA is complementary to this RNA, any piece is also complementary to this RNA, and this microRNA is simply something which can again find this RNA, bind to it, but when it binds to it, see what happens? It is complementary, right? So it binds in such a fashion now that ribosomes are unable to read it and you do not get the protein. Okay, and that's why it is referred to as mRNA or microRNA interference, or sometimes it's called as silencing RNA. Okay, or called RNA silencing. So what you are doing now is using a short piece of RNA to bind to mature RNA, and when that happens, that mRNA is non-translatable. Now, this happens naturally in our body, but again, people have made synthetic microRNA or what is called interfering RNA to target very, very specific protein translation, and shown that if you choose the RNA correctly, you can affect the way a protein is being produced by this complementary CRISPR. Okay, is that
But
clear?
so the whole
Yeah,
mRNA will be translated into protein, right? So why we are
correct.
targeting only small sections of the mRNA?
No, the point is, yeah, you are correct in the sense that the ribosome has to sit here and go all the way to the end, and I should get the protein. But the complementary RNA that I am making can bind to any small region of this, and when it does so, it will affect the way the ribosome can move, right? For example, if this short piece sits at this end here, then the ribosome will not even start. If it sits in the middle, the ribosome will start and get atrophied in the middle. I'll not get the protein. Or it binds to the end here. The ribosome will come all the way till this point, but will not be able to finish the protein. I'll get a truncated protein. Okay, so that's why for interfering with the functioning of this mRNA and to make the functional protein, I need not make an RNA which is totally complementary. I can just bind to a small region, but such that when it is bound there, ribosome is unable to transfer. Okay, and that's why you only need a smaller RNA. Don't need the full sequence. Okay. Are you seeing what I'm trying to tell you?
Yes, sir. Yes, sir.
Yeah. See, if I don't make a small part of the protein, also the protein will not function. Okay, and therefore my mRNA need not be as big as the original mRNA. Okay, or I should say my controlling RNA need not be as big as the original RNA.
Got it, sir. Thank you.
Okay. Okay. Let me go back. Let us see. There is something in the chat box. Let me see. Uh, okay. Let me begin. Why do we have some systems where gene regulation is done at the translation level? Won't it be easy to regulate it at the DNA to RNA level? Not necessarily. Okay. Uh, uh, Control at the transcription level and translation level are both equally effective and slightly different. Okay. Uh, and therefore, you can do so at both levels. No, what is normally found is that in bacterial systems, because many genes are clustered together and they're involved in the same process, to control at the level of transcription is much easier because I stop the expression of all the genes in one shot. But since in eukaryotes, that type of system is not there, and different places genes produce only one protein at a time, even though 4 or 5 may be needed for the same process, there you will find control occurs both at the level of transcription as well as translation. Okay. If I stop the RNA being translated, I can do exactly the same as RNA being made from dRNA. Okay. Why is it polycystonic? I just told you, bacteria don't have big genomes. So what they tend to do is use their genomes more intelligently. So all the proteins which are required for one process are typically found close to each other or right next to each other on the genome so that they can be produced together when needed. Okay, and therefore you find polycystonic messages more in eukaryotes. When lactose source is used up, then what happens to the allogalactose, which is bound to the repressor system? Okay, now remember, allogalactose binds to the repressor, and when that is there, the repressor is unable to bind to the sequence. But these interactions are all reversible interactions. Okay, it is not a substrate, it is not an enzyme or anything like that. It just sits there. The more the level of allogalactose, more will be the tendency for the repressor to be removed from the sequence. But as allogalactose levels falls off and galactose gets used up, lactose gets used up to galactose and glucose, then at that point allogalactose might be again converted back to galactose. Okay, because that enzyme can do both the reactions, and then that will again be converted to galactose and glucose, at which point the repression will start again. Okay. What is the difference between promoter and operator? They are very similar sequences. They all tend, a promoter, as they say, when things bind here, it enhances. An operator is somewhere where it doesn't indicate plus or minus. So you can have operator which will enhance and operator which will, even inhibit. Okay, but they are all sequences which are involved in regulating the way RNA polymerase will read the message on the gene which is right next to it. What is the role of spacers? As I said, spacers are pieces of viral DNA in the genome, increase the genome size and destroy the cell. Okay. Actually, in a sense, if that occurs in an unlimited fashion, yes. Okay, but what is seen is that there are some viruses which are able to enter the E. coli, and those viruses which tend to cause problems for E. coli are the ones which will be slowly cut and put into the genome. Okay. Now remember, these spacers are only about 50 to 60 bases. Okay. And remember, the E. coli genome itself is 4 million. Okay. So to make a dent in that 4 million size by adding small spacers during the lifetime of a bacterium, I wouldn't worry whether the genome size will become very big. And now remember, this even though this inheritable, later on when the virus is not seen for a while, then those spacers will be removed also. Okay, so I think that also occurs in the bacteria, that when a spacer utility is not there for a while, then the spacer actually might be removed, replaced with spacers which are more relevant to the viruses which are coming. In our body, while gene editing, we just introduce sDNA in the cell, but our DNA is supercoiled. Okay, so after identification of the required sequence, the one cell gathers help in one ending or Cas9 can. Apparently, it seems that Cas9 also has got this helicase function. Okay, so and that is something which is very interesting about Cas. That Cas can even go where DNA is fairly supercoiled, and it appears to be able to identify sequences and unwind the DNA. Okay, so Cas itself has helicase activity and also has nuclease activity. Okay, and after cutting, this Aditi is asking, the DNA repair systems come into play. Uh, so how is it decided that non-homologous end joining will occur or homologous? Okay. Now, when there is no other DNA to help, for example, if the cut is occurring in such a fashion that I do not have a template stand to take care of the problem in hand, then I have necessarily have to do non-homologous end joining. So I put random bases and somehow join it together. But if at that time I have got access to a sequence which can help me by acting as a template, then I can have homologous recombination. And when that happens, the error which will be less. Okay. In fact, this is what they do in gene editing using CRISPR and Cas technology. They actually give a homologous piece containing a mutation that they want. Okay. So not only is the target gene cut, but it is corrected and mutated to the form that you wish. Okay, and therefore, in Cas9 technology, not only you can cut target DNA, but you can actually modify the target DNA to get mutations that you wish to have. Okay, so I think that answers all the questions. Is there any other questions which are there? Any other questions? Anyone? Huh?
No, sir.
Okay. So let me see. I'm going to stop recording. Okay.